Hey guys, hope everyone's doing okay out there. We're going to get a start with something new this week, Chapter 10. We're going to be talking about DNA, and this week we'll be talking about DNA structure. So really the first part of Chapter 10 we'll be covering this week in this video. I'll also be sending you the PowerPoint for the entire chapter this week that you'll be using for the next couple weeks. However, this week we'll only really be covering that first part about DNA structure. So let's get started. When we talk about DNA structure, we often mention the two scientists, Watson and Crick, who were credited with creating the model of DNA. But it's important to note that there were also some other scientists who helped Watson and Crick, gave them good information to use to build their model of DNA. Those other scientists include Rosalind Franklin and Maurice Wilkins. Those two scientists were able to take X-ray photos of DNA to help get a good idea of what the structure actually looks like. So the structure of DNA was discovered in 1953. So really, not that long ago when you think about, uh, all the things that have come from learning about DNA, all the understanding of genes, and all the things that we use it for, coming up with different medicines, different things for taking care of people's health. 1953 was when we really found out what DNA was and what it looked like. DNA is a double helix. That means it sort of looks like a twisted ladder or a spiral staircase. And on a DNA structure, there are two chains of DNA that run up and down the structure. And this model that Watson and Crick created also helped us show how DNA could replicate or make copies of itself. We know that that's really important in each cell because before a cell divides, it has to make a copy of its DNA to give that new DNA code to another cell. So the DNA double helix is made out of two nucleotide strands that wrap around each other in the shape of that double helix, like a twisted ladder. Nucleotides, they're the monomers or the building blocks of DNA. And we're going to talk about some of their parts that make up those nucleotides in just a moment. But if you look at the picture on this slide, you can see the twisted ladder shape of DNA. On the outside, those light blue ribbons, those are made out of sugar and phosphate. So those are two very important parts of DNA. And then in the middle, we have these letters. You can see like G, C, A, and T. They're in different colors and they have different shapes. Those are called nitrogen bases. And there's 4 of them that we'll be talking about as we go through DNA structure. So as I said, DNA is made up of a monomer called a nucleotide. And in a DNA molecule, there's going to be just millions and millions, probably billions of nucleotides that make it up. But each nucleotide is actually made up of 3 little parts, and we can see it pictured in this slide here. The little circle, the pentagon, and the rectangle off to the side. Those are labeled there for you. The pentagon, that is the 5-carbon sugar that makes up a nucleotide. It's called deoxyribose. That is where part of the name for DNA comes from. DNA, if you don't remember, is called deoxyribonucleic acid. So deoxyribose, that's the sugar that makes up DNA, and that's the pentagon in our picture down towards the bottom. The circle in this picture is the phosphate group that helps to make up the sides of that DNA ladder, the backbone of that ladder, along with the 5-carbon sugar. And then the rectangle off to the side of the picture is one of the nitrogenous bases or nitrogen bases. And there are 4 different ones, and their names, we usually give them just a 1-letter name, like A, T, C, and G. But their names there for you are adenine, that would be A, guanine, which will be G, cytosine, which will be C, and thymine, which will be T. Now, we will be talking about another type of genetic molecule later on called RNA, called ribonucleic acid. And it's just a little bit different. It's not going to have the base called thymine. It's actually going to have a different one called uracil that would be U. So this nucleotide that you see pictured here, there are going to be billions of those that make up each DNA molecule. So when we look at the chemical bonds that hold DNA together, there's going to be two different types of bonds that hold it together. The nucleotides along each DNA strand are going to be held together by covalent bonds. So the sides of your DNA ladder that are sugar and phosphate, they're going to be held together by covalent bonds, which if we remember from earlier in this year, covalent bonds are types of chemical bonds where atoms are going to share electrons. Kind of makes them stick together like glue. So that's holding the sides of the ladder together. The rungs of the ladder are going to be made up of nitrogen base pairs that are complementary. That means they're going to link up in a certain way. And they're going to be bonded by hydrogen bonds, which are some of the same bonds that help to hold water molecules together and allow them to stick together. So they're not going to be quite as strong. We'll talk about how those bases, how those letters match up in just a moment. So those bases that make up the rungs of that DNA ladder, there's a certain rule that they follow for how they kind of match up and link up with each other in the middle of that DNA molecule. In the picture off to the side, you have a small section of that DNA ladder that's been untwisted. You can see the sides of the ladder in purple, and then the rungs of the ladder, they've been made bigger so we can see them, and you can see what letters they are that kind of hold together the middle of that ladder. If we look at this, we see a certain pattern. We see that A always bonds with T, so adenine always bonds with thymine. The way that I remember this is apples and trees. A and T always match up together. The other pattern that we see is that G and C always link together. So for C and G, I remember cars and garages. They always match up together. For cytosine and guanine. So these are your base pairing rules. A and T always go together in the middle, and G and C always go together. If you look at our picture, we don't have a G and an A going together. We don't have a C and a T going together. That would be a mistake that would happen in DNA if that were to occur. Usually it doesn't. So we can see also, and there's some dotted lines in this picture between those bases. There are three hydrogen bonds that hold together G and C. And there are two hydrogen bonds that hold together A and T. So this is really important because this helps make up the genetic code, it gives it organization, and it holds the middle of that DNA ladder together so our genes don't fall apart. There are two different categories for the nitrogen bases that we're looking at. The first type is called a purine, and those are the bases A and G. We can see them pictured together in a group down below. They are purines. They're called that because they have a double carbon ring. Okay, so they're going to be a little bit bigger than the other bases. T and C, the other bases, are going to be called pyrimidines. And these ones are only going to have a single carbon ring that makes up their structure. So when we look at how the middle of that DNA structure is held together, we're going to have one purine, so the two-ring shaped base, and we're going to connect it to one pyrimidine, which is going to be a one-ring shaped base, and that's going to be our pair that we have in the middle of DNA. If we don't have that combination together, those pairs are not going to fit inside of that DNA ladder. You can only have like a total of 3 rings going across. You can't have A and G linking together because that's going to give you 4 rings and make the ladder unstable. And you can't have T and C linked together because that's only going to give you 2 rings, and that's going to make the middle of that ladder not be connected all the way across. So we have to have that pattern being followed. So to wrap up this first section, we see our DNA model here shown in two different ways. In our cells, it's usually going to be that twisted double helix that's up on top. But a lot of times you might see it in a model that's untwisted, so we can get a better idea of how those bases match up. Look at that picture labeled B down there where it's untwisted. We can see that on one side of the ladder, it has a list of those bases. For example, up on top, it starts with A, C, C. If you notice, on the other side of that ladder, they're following those base pairing rules. A is matched up with a T. The shapes also fit together in this model. C and G always go together, cars and garages. So our next group, we have a C and a G, and if you notice, they fit together. After that, we have another C and a G. So look through that picture, we can see that those base rules hold true for that entire DNA ladder. A and T always match up together, and C and G always match up together. So sometimes you might be given maybe only the top half of that DNA ladder. Like you might be given A, C, C, T, G. And then you might be asked to write what's going to go on the other side of that ladder. So you would just use your base pairing rules to match it up and know that wherever you have an A, you're always going to put a T. Wherever you have a C, you're always going to have a G linked up together. So that gives us our genetic code that helps hold our DNA together, and that's something that we'll be using throughout the rest of this chapter. So make sure that you review that in your PowerPoint. Also, if you have your digital textbook code, I gave that code to you. You might want to make sure you review this part of Chapter 10 because that's going to be very important moving forward.